Okay. So we were talking about, in the recorded lecture, I was talking about how DNA codes for protein. And there's this little graphic that I actually use in my head that might be good for you to learn. And so this is the idea that DNA is transcribed into messenger RNA and then translated into protein. And then this is kind of fun. This, when I make a circle like this, you'll see this sometimes in the books. This is called replication. So DNA making copies of itself would be replication. This would be transcription. This would be translation. Okay. So there was what scientists called the central dogma of molecular genetics, and that was that one gene coded for one protein. So we had this idea that one gene equals one protein. And they called this the central dogma, which essentially means that this was just like such an ingrained idea that it was really hard to get away from this idea, okay? And so with the Human Genome Project, they discovered that we have about 20,000 genes, right, in our genome. That is far fewer than the number of proteins that we have. So we have what is referred to as the proteome, which is about 100,000 proteins in our, they call it the proteome, kind of taking off on the idea of genome, right? So this would be all the proteins, and we know that proteins are super important because they're the ones that are biologically active molecules. So they're the ones that are enzymes that produce the carbohydrates or break down the carbohydrates or produce the DNA, right? So the enzymes that allow the DNA to be replicated, like DNA polymerase, those are all proteins. The proteins are what's significant. So now we're trying to figure out what our proteome is, all of the proteins in the body, so they have, an encyclopedia of them. And then we're trying to figure out how only 20,000 genes could code for all the proteins in our body. And so what they discovered is this is not true. Okay, so I could put a little cross between my equal sign there, that one gene can actually code for more than one type of protein. So this is done through what is called messenger RNA processing. So this takes place in the nucleus after transcription. And we all, or geneticists all, thought that genes were all the DNA, the sequences that coded for a protein in a gene were all together. But what they discovered is that it actually has some sections which are non-coding, which have to be edited out of the transcript, which is the messenger RNA. So we have exons. So these are sequences of A, C's, G's, and T's that code. So these are coding sequences. That means that they are important for the production of the protein. Then we have what are called introns. And they're called introns because these are intervening sequences. So if you can think of intervening sequences that have to be cut out, then you might remember that introns are the ones that are cut out, and the exons are the ones that are kept. Okay. So we have introns, and these would be non-coding sequences. Okay. So somebody visualized this as being like a piece of film like in the old type projectors. And so when you wanted to splice something out, you would cut it out, you would splice it, and then you would put it back together. And so there are enzymes that are called spliceosomes. So spliceosomes are enzymes that cut introns out and splice exons back together. Spliceosomes. So here you can see in this diagram, the primary RNA transcript has introns in it, then it's spliced, those introns are cut out, the exons are put back together, and then the messenger RNA leaves the nucleus and goes out into the cytoplasm. So we have a phenomenon where the exons can be put together in any order, in any combination. And so this is what is referred to as exon shuffling. So if you imagine, if we're going to cut them out anyway and put them back together, maybe we could put them back together in different orders, right? So exons are put together in different patterns, we'll put. Okay. So for example, you could have E1 put together with E2 and E3. So those are 3 exons, right? You could have E2, E1, E3. And here they even show that some exons could be removed. So you could get rid of like exon 2, and you could splice exon 1 and 3 together, and that would be produce a protein that is shorter than this one, right? You could even maybe, well, we don't need to know. We could retain an intron in here. This is a little bit more complicated than you actually need to know in this diagram, okay? But that just means that we can put them back together in any order. So what this means is that one gene can actually code for more than one protein, okay? So that just overturned our central dogma of molecular genetics. And they don't exactly know, you know, how this all goes together, because they know the sequences, but they don't understand all the patterns of exon shuffling and how it could possibly produce all the different proteins. So you can see how this is going to complicate things immensely when we can't just say, oh, here's a gene and it goes exactly for this protein. They don't match up one to one. I don't know why they do that. It doesn't quite make sense. It seems like you should just have more genes. Why don't we just have more genes? Yeah. I'm not sure why they do this. And for some reason, I think it might not occur in bacteria, but I'm not sure. I have to look that up. I think this might be a eukaryotic phenomenon, but I'm not sure. Okay. Okay, so let's look at protein synthesis. So remember that this takes place in the cytoplasm. So this occurs in the cytoplasm at a ribosome. And remember that a ribosome itself is not an organelle, but some ribosomes are attached to the endoplasmic reticulum, and they are called the rough ER, right? Some ribosomes are just floating around in the cytoplasm. So in order for protein synthesis to take place, we have another type of RNA that we need to talk about, and this is what is called the transfer RNA. This is abbreviated as the tRNA. So we had messenger RNA or mRNA. This is completely different. It serves a completely different function. The tRNA is our translator. So this is my translator. So it has what is called an anticodon. So this is a 3-base sequence. that is complementary to the messenger RNA codon. So it's called the anticodon. And then it also has an amino acid binding site. Okay. This, unlike messenger RNA, which can be really long, messenger RNA can be thousands of bases of A's, T's, not AT, but A, U, G's, and C's, right? tRNA is very small. So it's a small molecule. And they just float around in the cytoplasm. Near ribosomes. So they're not really attached to the ribosomes, they're just kind of floating around in the cytoplasm near the ribosome. So if we look at the diagram that shows the interaction between messenger RNA, ribosomes, and tRNA, it looks like this. Okay. So here, you need to know this diagram. So this is the diagram that you need to know. If I took out all the little captions, you would be able to tell me what these are. Okay. So these are the subunits to the ribosomes. The ribosome is protein. So just think these are the two subunits of the ribosome. This is my messenger RNA that has been processed and has left the nucleus and come out into the cytoplasm and attached to a ribosome. This is my tRNA. It's a smaller molecule. This is my anticodon, and this is my codon. Okay, and then this would be my growing chain of polypeptides, which are many amino acids bound to one another. Okay. So when I look at my tRNA, I can draw my tRNA. It might look something like this. Okay. And I can draw my messenger RNA, and it might look something like this. Okay, so this is my messenger RNA. And oops. And so that's just a squiggle. And this is my tRNA. Okay, and it's bound to an amino acid up here. Okay, so notice how the tRNA can speak both languages. It can speak amino acid language, and it can speak nucleic acid language, so that's why it's the translator. So tRNA, just like messenger RNA, does not have any T's, does not have thymine in it. So we could say that this would be, for example, U, A, G. Okay, so that's my anticodon. So using the complementary pair, U would pair with A. A pairs with no, U. And G pairs with C. Okay, so then I forgot to bring my table with me, so I Google it. I Google the genetic code because that's an easy way to find my table. Remember that I will give you the table, right? And it might look something like this. Just the 3 letters. So I will give you a table and it will look like this. So what was it? It was U, E, U, A, C, right? UAC? Okay, so that's ILE, although it looks like 2 L's. But that's isoleucine, but you don't use the name. It's AUC? Yeah. UAC. Okay, sorry. UAC.
The mRNA or the
Messenger RNA. The messenger RNA is what?
it was
AUC. AUC. Isoleucine. Okay, so just put ILE. That's the amino acid. ILE. Okay. this is, you can think of it like a factory, and this is like a conveyor belt, right? So as the amino acid is brought in, it is added to the growing chain, and then this tRNA leaves, and then a new tRNA comes in, and it brings with it the next amino acid. So you could Google, you know, translation, show me a video of translation, and there's lots of videos out there that show it animated, showing it as kind of a factory process.
Is this anything like the little circle one, the table?
Do you have the circle one in your book?
No, I used it. No, I was just wondering.
So if there is a circle version of the table.
I've used that one.
Yes, it's the same
Similar
thing.
to the table?
It's the exact same thing, information. It's just read differently. I haven't, you know, when I Google it, it doesn't come out with that. Circle. So some books, it looks like this, which is kind of cute. Right. Yeah. So you guys have seen this one? So this is is this the first letter, second letter, third letter? So A, where's AU? Oh, geez. Isoleucine. There is no AU, that's weird. It's a weird one, let's look at this one. Oh, I can't see it. Anyway, but I'll give you the table, not the circle. Okay. Okay, any questions about this idea? tRNA is very different from the messenger RNA. Okay, so this is an overview. So remember that, again, transcription takes place in the nucleus. We produce a messenger RNA. The messenger RNA is processed. So the introns are cut out, the exons are put back together, and then it goes out into the cytoplasm to be translated into a polypeptide. And what a polypeptide means is that it's just a chain of amino acids. And then that is going to fold into the complex three-dimensional structure that becomes a protein that is capable of doing things, right? So what is it called when a protein unfolds? Denaturation, right? So denaturation is when the protein unfolds and doesn't work anymore. And it's really hard to get it to renature, right? It's almost impossible. Okay. Okay. So remember that was a description of how the genetic material could code for differences, right? So we talked about differences between individuals being due possibly to a protein, a difference in a protein. So the third thing the genetic material must do is it must undergo rare changes. And so these are what are referred to as mutations. So mutations occur at the genetic level, and they are random. So this is random changes. In the sequence of DNA. So it's very important. There's a misconception that you can mutate towards a goal because that's what happens in science fiction movies, right? But that doesn't ever occur in real life. Mutations arise randomly, and if they are beneficial, they are selected for. If they're bad and deleterious and maybe even lethal, then they die out, right? But mutations, as we learned last week, are extremely important because this is the way we get new genetic material, okay? So it's a mechanism for getting new genetic variation. And it actually might be material too. Maybe we'll talk about gene duplication. Sometimes genes get duplicated and one goes on serving its normal function and then the other one takes up a new function. And that's how you can get a brand new gene appearing in a genome of an individual. Okay, so when we talk about mutations, we can talk about what are referred to as point mutations. So this is just a change in a single base. And it is a substitution. So you substitute an A for a G, right? So you could substitute, you could have A, G, right? And then that could turn into G, A, G. Right? So we substituted this A, we substituted a G for that A, and that would be called a point mutation. And so the one that causes sickle cell anemia is an example of a point mutation. So sickle cell anemia is an example of that. Okay.
So
Oh, go ahead.
in point mutations, it doesn't happen when they're transcribing replicating and it swaps it, or?
It can happen during, so when I'm talking about the DNA, it would be during replication that it happens. So when the genetic material gets copied, a mistake is made. And we'll talk about things that can cause mistakes to be made in a minute. Okay, so we have point mutations, and then we have what are called frame shift mutations. And these are generally much more dramatic because they are an addition or deletion of bases. And we kind of talked about how on the Creed chat, that was the deletion of a whole part of a chromosome. So when the chromosomes lined up and they paired, the chromosome broke, right? And so that'd be a huge deletion. But sometimes we just could get a deletion of a single base, right? And so to show how or why it is called frame shift, I'm just going to give you a sentence which reads, the cat ate the, but this is in capital letters too. The cat ate the rat. Okay, so notice how these are in 3, like the codon, right? So these 3 letters make up a word, right? But imagine if I deleted one of those letters out and I shifted my frame of reading. So now it will read THC ATA TET HERAT, right? And that is gibberish, right? And so what it means is that the frame shift mutation could cause really dramatic effects in the protein, and so they are much worse, and so you probably would be selected out if they happen because they cause such a dramatic effect, right? That's why it's called a frame shift. Because it changes the reading frame, how the genetic code is read. Okay, so these are kind of types of mutations, and then I want to talk about causes. Okay, so I'll put causes here. Oops, okay. Okay, so causes, one cause is what is referred to as spontaneous mutations. And this is where there is no external factor causing the mutation to arise, it just happens. And this is due to the instability of the DNA bases. Instability, that reads instability. So the DNA itself, the bases itself have a built-in instability, and they exist in 2 forms, and these 2 forms are called tautomers. Okay, so this is 2 forms. One form of the base is very stable. So it exists in this form 99% of the time. So 99% of the time it is in its stable form. and then it wobbles, and it becomes it goes to its unstable form. Okay, they call it wobbling. I have wobbles. So it's just like, and then it's a little bit different, and then it shifts back. So it kind of shifts back and forth. And so if it's in its unstable form during DNA replication, you could get a mutation arising just because the genetic material spontaneously mutates occasionally, right? That's one of the things we said it has to be able to do when we were looking for the genetic material. It has to be able to, on occasion, mutate or change, right? So, cytosine normally pairs with what? Guanine. So that would be in its stable form. If cytosine is in its unstable form, it's going to base pair with adenine. So when the DNA polymerase is reading the code and making the template, and using the template, excuse me, to make the new daughter strand, it's going to make a mistake there. Okay, so let's just look at, you do not need to know what these tautomers look like. I'm not going to make you memorize them or anything, right? Oops. So this is cytosine in its stable form. So the top one is in its stable form. Okay, so notice like there's only one covalent bond here. In this one, there's two, right? Notice that there's no hydrogen here. In the unstable form, right, there's a hydrogen, right? So in this case, cytosine mispairs with adenine, right? And voila, we have a new mutation. So that's what's called spontaneous mutations.
So what does that cause?
That could cause anything. So we typically, they estimate we are born with 50 novel mutations in us. So that is mutations that are found in us and nowhere else. Because you have to remember that your genetic code is 3 billion base pairs in length, and most of it doesn't code for anything. So there's going to be some changes, and it's going to have no effect, right? But if it changes in a protein coding gene, then it could have a good effect, it could have a bad effect, or it could actually have no effect. We'll talk about that in lab too. Okay. Okay, so this is different from what we call induced. So spontaneous mutations are due to tautomers. Induced mutations are due to things that cause mutations, which are called mutagens. So if you think about this word, that means mutation generators. Okay, they generate mutations. So some examples of this would be radiation. So sunlight, UV radiation. That should be a, let's see, that doesn't look right. UV radiation. Okay. And even if you do tanning beds, right, you're subjecting yourself to UV radiation, and you could possibly get mutations from tanning beds, right? They shouldn't allow tanning beds, in my opinion. But, so sunlight causes a specific type of mutation, which is called a thymine dimer. So what happens is that the thymines that are next to one another, instead of complementary pairing with what they should be pairing with, they pair with each other. So it kind of looks like this, like that. So instead of having hydrogen bonds with their adenines, they produce a covalent bond with one another, and it actually causes a kink in the DNA. And if this DNA then separates to be replicated, then when it gets to here, it's not going to know what to do, and it's going to skip it, right? So a mutation can happen. It could be a frame shift mutation, because you would have deletion of those bases in the new strands, right? So luckily, we have an enzyme that can repair this, and it's called photolyase. So this is an enzyme that repairs I'll get that for you. Repairs thymine dimers. So it can go around and edit some of those out, but sometimes it doesn't do a very good job, and you can get mutations that accumulate and can lead to skin cancer. So this can lead to skin cancer. Now, there is a genetically inherited disease where people cannot edit out the thymine dimers, and so they accumulate mutations really rapidly when exposed to UV radiation. And so these are the people and the children that cannot go out during the daytime or cannot be like next to a window where there's UV radiation coming in. And they have a disease which is called xeroderma pigmentosum. Yeah, so it makes you more sensitive. So some, yeah, some antibiotics.
So does the thymine dimer lead to skin cancer, or the?
It's the mutation that causes. So when that is red, a mutation will happen.
Okay.
And then it can, if that is in a important gene regulating.
From the thymine dimer.
Right, right. So if we look at this, there is pictures that show this. Oops, that's not what I wanted to show. Okay, so this is the thymine dimers, right? That's my thymine dimer. This is it repaired. So we have an enzyme that repairs it. If that doesn't happen, then you have people that even exposed to sunlight in brief periods, so like maybe this person wasn't out in the sunlight for more than 20 minutes, right? And they start to develop sores on their skin. And some of those sores can become skin cancer. And so they just are super sensitive. And there's not very many of these people around at any given time because it's a rare mutation. But they do have camps where they bring the children together and they play at night outside and then sleep during the day so that they're able to get some interaction with people, other people that can't be exposed to UV radiation. Okay, so this was another example. Does anybody know who this is? Probably was in the news. So this is an example of polonium radiation. So polonium exposure. So polonium is like uranium in that it's radioactive, right? And so it can lead to radiation sickness, right? Radiation illness. So this was the ex-KGB officer from Russia who went to the United Kingdom to get away from being, I don't know why, he was in trouble, right? So he went to the United Kingdom and they say that some Russian people came into the UK and slipped him some polonium and he got really sick and he died. So this was supposedly a Putin example of the Russian president killing off people that he didn't want being out of the country and telling KGB secrets. So that would be radiation. Sometimes in the military, they use uranium or spent uranium and they put it in the ammunition. And so that has a little bit of radioactivity too, and that would make people very sick if they were shot with bullets that had uranium in them. Okay. We obviously also have X-rays. So X-rays today are much safer than they were in the past. So Madame Curie, the woman who discovered and used X-rays extensively, died of cancer, right, at a relatively young age because she was using X-rays that were very, very strong and they were causing damage to the DNA. Okay, so we also have what are called chemical mutagens. And chemical mutagens can act in a variety of ways, but essentially they change the bases. So they change the structure of the base so that it can possibly bind with the wrong complementary base. So I'll put that can change the structure of the base. It pairs, we'll put, it pairs incorrectly. So probably the most common chemical mutagen that most of us are exposed to are nitrites. So this is in preserved meat. So like salami, like you guys probably gonna go home and have like salami and cheese and crackers, right? Full of nitrites. Okay? And so nitrites actually change the structure of cytosine, for example. So instead of pairing with guanine, it actually will pair with adenine. So if we look at a picture of how this works. Oops. So this is not from your book. This shows nitrites, sorry. So this is where cytosine normally pairs with guanine, but it changes cytosine into uracil, so it actually pairs with adenine. So instead of cytosine pairing with guanine, cytosine pairs with adenine. It actually becomes uracil. But anyway, so this would be, so sometimes I think it's important that you don't let your kids specifically eat a lot of preserved meats, and specifically if you're buying hot dogs, it's probably a really good idea to buy the hot dogs that do not have the nitrates in them. So if they're like, they eat, like if that's their favorite food, then that can be a real problem, right? They don't. This leads to digestive problems. So it's linked to people maybe having colon cancer later in life, stomach cancer, right? So cancers of the digestive tract. So a lot of chemical mutagens are also carcinogens. They're not all carcinogens, but they can also be. So we can also be carcinogens. So carcinogens can be other things than chemicals, but most chemical mutagens do eventually or can eventually lead to cancer. So that's why they're called carcinogens, cancer-causing chemicals. Okay, tobacco smoke, for example. So example would be cigarette smoke. Okay. Is carcinogenic because it has chemicals that are mutagens in it. Okay. So let's talk about the types of mutations. So we can have what are called somatic mutations. So a somatic mutation means that it is in a body cell. And it will maybe, not necessarily, it may not be passed to the next generation. So this is in a body cell. And this is versus germline mutations, which is in cells giving rise to egg and sperm. So a germline. So mutations that happen in my skin or happen in my stomach as I eat too many preserved meats are not going to be passed on to my offspring because they are not in the cells that are going to be passed on. That's why when you go in for x-rays, they protect your ovaries and your testes, right? They want to protect those because those are the most susceptible to germline mutations that can be possibly passed on to the next generation. And usually mutations that are caused by that would lead to inviable sperm or eggs that are inviable and could cause problems with fertility. Okay, so we can also talk about, so this is another type, we can talk about inherited versus sporadic. Okay, and inherited would be germline, right? So these would be mutations you would inherit from your parents, but sporadic mutations occur in the individual. These are passed to you from your parents. Okay. So some cancers have a inherited predisposition. So you can have an inherited predisposition to getting cancers. So they tend to run in families. So we're going to look at this next week in lab when we do our DNA fingerprinting. And some cancers just arise, like some weird mutations just arise sporadically. And so I could get a mutation and I could get cancer, and it could be not of the inherited kind, right? So you can get traits that are not inherited. So a good example of that is Lou Gehrig's disease or amyotrophic lateral sclerosis. So only about 10% of ALS people receive it inherited, and other 90% is a sporadic mutation that causes that particular disease. So it's important to realize that not all genetic diseases are inherited. Right? So I'll put that in writing. Not all genetic diseases are inherited. Okay.
If you develop a sporadic mutation, does that necessarily mean that you can pass it on to future generations?
But it'd have to be a sporadic mutation in your germline cells.
Okay.
Yep. Yep. So maybe you had a sporadic mutation when you were an embryo, and then it got passed down to a lot of the cells in your body.
Okay.
Yep. Okay. So the next thing we're going to talk about is how genes are regulated. So when we talk about gene regulation, we oftentimes say a gene is turned on and a gene is turned off. Okay. So turned on means that it is producing protein. Turned off means that it is not producing protein. And this gene regulation is really important during embryonic development because this explains how cells specialize, right? So explains how cells specialize, specifically during embryonic development. Because all of my cells have the exact same set of genes, but my skin cells are going to be dramatically different from my neurons, and they're going to be dramatically different from my muscle cells. And so the regulation of gene expression is the whole story, really, of how cells become very specialized and only produce certain types of proteins. So some genes are turned on and some genes are turned off. Okay, so one way that gene expression is regulated, one mechanism is through how the DNA is packaged. So we're going to talk about packaging of DNA. So does anybody remember what type of protein is DNA packaged with? Does anybody know? What are the proteins called that wrap the DNA? Histone proteins. Okay. So DNA is wrapped around histone proteins. And when it's not being, when the cell's not dividing, the genetic material is chromatin, right? So the chromatin is even packaged so that some parts of the DNA are wrapped more tightly and some parts of the DNA are wrapped more loosely. So we'll say that if it is tightly wrapped, this is not coding. It is turned off, and it is called heterochromatin. It is also darkly staining. So it's wrapped so tightly that DNA or RNA polymerase, RNA polymerase can get in to actually read and transcribe the DNA into messenger RNA. It's very simple, just wrapping up the DNA. Okay. If it is loosely wrapped, it is generally coding, which means that it is turned on, and it is called euchromatin. So what does U mean again? Means good. So for example, I'm a euploid, I'm eukaryotic, right? I have euchromatin, right? That's good, okay? So this is lightly staining. Because there's not as much DNA in that particular area because it's loosely wound. It doesn't pick up as much stains, so it is lightly staining. Okay. So if we look at an image of this, okay, so this just shows the DNA as it, this is not from your book, but this is as it's wrapped around the histone proteins. And then eventually, if the cell is going to divide, that chromatin, which kind of looks like this, right? So this is my chromatin. This is heterochromatin so, because it's so darkly staining. My chromatin is eventually going to be packaged into chromosomes. And so if I look at a chromosome, so this is my chromosome, and I'm going to look at where it is darkly staining. Right? So what's the end of the chromosomes called again? Telomeres. The telomeres don't code for it because we lose telomeres, right? As the mitotic clock counts down how many times mitosis occurs. So the telomeres generally stay dark. Okay. The centromere, the same thing. That is a place where the cytoskeleton attaches to pull the sister chromatids apart. So generally that is not where we have coding DNA. And so where it is darkly stained, so I'll put a point right there, like there, that is heterochromatin. Where it is lightly stained, that is euchromatin. Okay? So where there's not a lot of stain, it's euchromatin. Okay? So we can kind of identify where in the DNA we have genes turned on and coding for protein. Okay, let's see, what else do I have? I think I'm probably going to need to stop there. Oh, here's another really cool picture. So this is actually an electron microscope picture of the nucleus. So this is an actual picture taken with a much higher powered microscope than the light microscopes we use. And you can see that the chromatin, there's some areas where it's lightly staining and some areas where it's darkly staining. And that has all to do with the way that it is wrapped around the histone protein. Okay, so I'll stop there for today. And we won't have any additional lectures. So you want to focus on doing your extra credits over Thanksgiving. Okay, your extra credit is on the very top of your Canvas page in the very first section. That is where you'll upload it and submit it.
When is it due by?
You know, you'll have to look. It's got to be something beyond that we talked about in class. So you couldn't just like summarize your notes. You actually have to do a little bit of research. Yeah. You know, I have to go downstairs and get it. So if you want to wait a moment. I was hoping to print it from here, but the printer is not set up on this